My name's Nick Zuchowicz. I'm a PhD student in mechanical engineering at the University of Minnesota at the Bioheat and Mass Transfer Laboratory. We work with a couple of species of fish embryos, we work with a couple of species of shrimp, and my specialty, corals. We're interested in cryopreserving the embryos and the sperm and the symbiotic algae that live inside the corals. This right here is called a mushroom coral, and this is the skeleton. In life, they look like this. You can see why they're called mushroom coral. Those are little juvenile corals that are going to fall off of that aggregation and turn into these little free-living corals. When they spawn, they look like this. Here you see a mushroom coral spawning eggs. They can be male, they can be female. In this case, you're looking at a female, but they will also spawn sperm, which will look very similar, but cloudier. I'm really interested in cryopreserving the sperm and finding methods to do that effectively. Generally, when we cryopreserve things, we use one of two methods. So either slow freezing, which you see on the left, or vitrification, which you see on the right. In slow freezing, we intentionally form ice in the sample, and that creates an increasing concentration of cryoprotectant chemicals in the sample, which eventually turns to a glassy state with the cells inside. On the other hand, in vitrification, we're looking to form a glassy state throughout the sample, both in the liquid phase around the cells and also the cells themselves. The other folks who are going to show you things on this tour, they work on vitrifying organs. For my purposes, I'm mainly interested for this project in slow freezing, so intentional ice formation to promote cryopreservation. This device that you see in front of you has gone through a number of iterations. An older system, this floating rack, that has some problems with reliability, with cooling rates, which we'll see in a moment. There's the system that you see in front of you, which is widely used by coral cryopreservation practitioners throughout the world. And then on the right, you'll see this more high throughput system that can freeze 48 vials at once. So there are a number of different versions of this system. With the older system, the floating racks, you'll see that there's quite a bit of deviation between the various samples and from run to run in the cooling rate. Whereas with this newer system, we get pretty consistent cooling rates, so plus or minus 1 or 2 degrees per minute. I'll give a quick demonstration of how this system works. You have a dewar that contains liquid nitrogen. You have this frame system that allows you to check the level of the liquid nitrogen on this gauge right here. This is placed in the vessel, allowed to thermally equilibrate. We then have a number of samples here on this insert, and each of these would contain about a milliliter of sample. Once this system is equilibrated and the liquid nitrogen is at the correct level, this insert system is dropped in, the lid is placed on, and about 6 to 7 minutes later, you have a cryopreserved system that you can move into storage. In spring 2023, we also brought in a senior design team, so that's fourth year mechanical engineering students, who worked on an improved system, one that has active rate control and feedback. They got a chance to work with some of our collaborators, specifically Mary Hagedorn, who's out in Hawaii and pioneered the field of coral cryopreservation, and Dr. John Daly, who runs the Reef Restoration and Adaptation Program's cryopreservation program. We also have other collaborators who make use of this system in Mexico and in Curacao. Ultimately, when we freeze down coral sperm samples, these are destined to go to the National Animal Germplasm Program in Fort Collins, Colorado, a large repository. We have millions of samples across many species, and this is where our coral samples end up. These samples are also useful in the near term. So we have used frozen sperm for long-distance transport of genetic material from one population to another across the Caribbean, showing that cryopreservation is not just something for the long term, not just a hedge against extinction, but in fact allows us an active management option for genetics in genetically depauperate populations, that is, populations that have become very genetically restricted. So we're enhancing resistance to climate change by moving genetics around, something enabled by cryopreservation.